From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 18th, 2024. Hello, Micronauts, and welcome again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 60th episode of Matters Microbial. I really hope to continue to do this podcast in the future, so your listens, views, comments, suggestions, and questions really matter to me. Last week, as the podcast was recorded, I spent some time in the microbiological mecca that is Yellowstone National Park. As I looked across the microbial mats, I saw a version of how the early Earth must have appeared. There were boiling springs, as seen here. Please note that how, as the water cools, the microbes become apparent and have beautiful colors. There were even tiny geysers to match the larger ones, as seen here. This particular hot spring runoff shows again how microbes need to balance the temperature and mineral content. Notice also how iron oxidizers create plumes of iron visible to the eye. It was a remarkable visit, and I was very fortunate. In my Bio 350 class at the University of Puget Sound, my Micronauts celebrated International Microorganism Day, as this logo illustrates. One student created a rock band of microbes in celebration. Another student wrote a haiku to their yogurt, something I recommend we all do. Microbes truly do rule and are indeed the basis of the very biosphere. And in the final analysis, they are first evolved, last extinct. At this time of year, I'm encouraged to get an influenza vaccine. Influenza is a wily viral beast and ever-changeable. Some of that has to do with errors in replication of its genome, as well as recombination. It is a beautiful, mysterious, and genuine health challenge. So it's a pleasure to introduce Dr. Adam Lauring, MD and PhD, to discuss the work of his research group uncovering issues of viral mutation, immunology, and how that factors into our experience with influenza. Welcome to the Quality Quorum today, Adam.
Thanks. It's great to be here.
It's really nice. I've often been told that we need to have more virology on the program, so here you are.
Yeah, I've seen you've had some phage people before. We consider them part of the viral crew. And so, yeah, it's great to be here to spread the word that there's other microbes.
And some of which, as we see all around us, impact our everyday world. I'd like to kind of start, if you wouldn't mind, because having an MD and a PhD may be something that some of the listeners and viewers aren't familiar with, the idea of being a medical scientist. Would you care to discuss that for a moment?
Sure. So the idea behind the MD PhD is, I guess, probably years ago, the NIH and the community decided that it was going to be important to have medical scientists who are trained both in clinical medicine as well as in how to do science. And so they set up these programs that are supported by the NIH and other funders to train people. And I was interested in both aspects, both kind of basic science, basic investigation, as well as clinical medicine. And so it seemed like a natural path for someone like me, and it's been great. So I did my training up I-5 from you at University of Washington in Seattle, and was there before Amazon became big. And that was a great experience, and I still to this day do clinical medicine. I do infectious disease work, seeing patients with infections, not just viruses, all manner of infections. And then I spend most of my time, though, running a research group that studies how viruses work.
I have to tell you, the physicians that I deal with, when, for example, when my late brother was so ill and they brought in ID docs to talk to me, they were so disturbed that I was a microbiologist talking to them, and as if I was going to somehow do something wrong. It was pretty interesting. But can you imagine how they would have felt if it would have been an MD and PhD talking to them?
Fair enough. Yeah. Fortunately, the name tag that I wear in the hospital just says MD on it, so I can kind of go incognito. And most people don't know.
It's also, I wanted to ask you about this question because I get students who ask me this one. Do you have to have an MD to do medical research?
No, no, you don't have to have an MD to do medical research, and you don't have to do a PhD to do medical research. The important thing to do medical research is just to have training in how to do science and an interest in problems that are related to clinical medicine, and gain the experience through doing. And yeah, so I've known people who are very successful doing medical research who have only MDs or only PhDs, and I think it's a credential, but really it's what you do and what you're interested in that shapes it.
Yeah, I used to tell my parents who didn't really know much about academia, it's like a union card almost.
Yeah,
And it is a good way to think about it.
right.
You're good people, bad people, the
Yeah,
whole gamut, just like any other job.
yep,
There's nothing special about the white coat.
right, for sure. Whether
But
the
I
white
thought
coat has media stains on it or blood stains on it.
actually nowadays media stain is kind of a pun when we think about it. So
Right.
that's actually well stated.
Yeah.
I also want to say that it's really important, especially when we talk about human impactful science, how important it is to do outreach to the public. And I know
Yeah.
you've done some of that on your website. You
Mhm.
have a whole section where you have particular articles that you've written for well, I guess they're not newspapers anymore, but you know what
Right.
I'm getting at.
Yeah.
And I'm going to have a link to those because I
Great.
do think part of what we all need to do in the microbial science is outreach to the community.
Yeah.
I call it microbial literacy, which certainly isn't original with me.
Yeah. Well, I mean, obviously, people, you can see, I mean, a lot of this has been, from the COVID pandemic, right? Where there was such a need to explain the science. And I think, I've always thought it's important to to explain and to teach for all audiences because, and for the general public in particular, because when you get right down to it, they are stakeholders in what we do and they support our work through, government and other funding. And so, you know, we need to explain what we're doing and teach so that, society's better.
I don't know who first said it. I've heard it attributed to a lot of people,
Yeah.
but if you can't explain what you do, I'm going to use the expression to a non STEM English major,
Yep.
then you don't really understand it yourself. And there's some truth to that.
Yep.
So I make sure that my students are always able to do that.
Yeah.
And it's good that in everyday life, when we talk about our science, we're able to do that as well. So thank you for your contributions to that area.
Oh, well, thank you.
I think it'd be a good time now to talk about the research interests of your group, which really
Yeah.
struck me because I'm interested in variation and mutation a good deal myself.
Yeah. Yeah. So my work is all about virus evolution. And when I think it wasn't a linear path to what I do, um, I just have always followed whatever I'm most interested in at the time and try to do it and try to get funding to do it. And But kind of looking, to kind of one through, a theme that runs through it is try and understand how and why viruses make mutations and what that means for how viruses behave or spread or make us sick. And so we started off just understanding viral mutation rates and what they are. Why do the viruses that I study make so many mutations, and is it a good thing or a bad thing for them? And then we started looking at the consequences of mutations, and then of course, because viruses make mutations, that makes them a little more difficult for us to combat and control. And so trying to understand those aspects, which I'm sure we can get into in depth.
I have a saying that I would love to take credit for, but I'm sure other people have said it.
Yeah,
It's the hand of Darwin is on everything that lives.
yeah,
And even though viruses aren't strictly speaking alive,
right,
they're life adjacent. How about that?
sure,
Certainly, evolutionary things happen to them,
yeah,
effects on them. And so when I work in the laboratory and I make a mutation in a bacterium to become resistant to an antibiotic, so there's a lot of thought about where that mutation needs to occur to create that change in what we observe, the phenotype.
right,
And you are basically have to do the same thing with viruses.
right.
Could you tell us something about how you do that?
Sure. I mean, when I started out on this work, um, we there's kind of two approaches. One is you kind of like you're saying, look at the phenotype and and then trace it back to the genetic lesion, right? And so, or you can follow it forward and do what's called experimental evolution. Um, and I see you've had several of really great experimental evolution people working in bacteria who've been on your show. Um, and and then obviously the whole field with, Rich Lenski and the long-term evolution experiment. And so you can, evolve pathogens in the lab and see, what they do or provide certain, selection pressures to see how the pathogen will respond. And so we make use of those techniques as well to try to understand, um, uh, you know, the forces that are acting on a virus. And the nice thing about viruses is, well, they evolve very quickly. And so you can get a lot of generations in a short amount of time and really see a lot of change. Um, and kind of speeds up uh, the process of discovery. And that's why viruses and phage have been excellent model systems for understanding kind of the fundamentals of how evolution works. Contributed not just to our understanding of viruses, but an understanding of evolutionary principles and things you can't do for other systems, experimentally, right? You're stuck with the fossil record and things like that. And so it's in a powerful system and it's fun.
I want to just basically say
Yeah,
something really important to listeners and
yeah,
viewers is that modern molecular biology, which primarily worked with microbes, it's true though,
yeah,
was pretty much populated by former physicists.
yep,
Now, the reason that I bring this up is that I really do believe that different thinking, different faces, different voices, different
yeah,
brains really help what we do in
right,
science. And that's a wonderful. You mentioned the Luria Delbruck experiments, for
yeah,
example. I think you have a beautiful picture
yeah,
of the two people enjoying themselves.
yeah,
Uh, certainly Max Delbruck was a quantum physicist, and that kind of thing is
yeah,
so important,
yeah,
especially when there's a fast moving change.
yeah. Yeah, and you refer to this image in this slide of Lurie and Delbruck at I think they're at Cold Spring Harbor. It's a famous photo. I'm not sure where I found it. Probably Googling. And but I think it's just I love it because here are these two giants who really figured out some of the fundamentals of mutation. And they're just sitting. I think it's Luria with his shirt off, and Delbruck just like take it in the afternoon sun, and it's just great. And yeah, their work is transformative. And yeah, Delbruck and so many others, as you said, were schooled in physics, and it brought a new perspective and a quantitative perspective, I think, which was so important early on. And then you know, the other thing to note is, for your audience, is a lot of this work was done in lambda phage and also, in, bacteria, and trying to understand, how this worked. And so it was microbiology from the start to try to understand kind of these basic life processes.
And relevant to today's discussion, it's really critical for listeners and viewers to appreciate that mutations are essentially random.
Right,
And it's very counterintuitive to many,
right,
many people.
right,
And so, for example, if you have a watch and you put the
yeah,
wrong size piece in it,
yeah,
it's not going to work
yeah,
at all. So this confuses many people when we talk about mutations. And in fact, they're happening all the time, as you mentioned. The
right,
question is whether or not any of the results of that have any advantage in terms of natural selection.
right. The
So,
oh, go ahead. Sorry.
oh no, you were going to finish your
Yeah,
thought.
yeah. I mean, in fact, Lurie and Delbruck, their fundamental contribution was to show that mutations are a chance process. It's random. Um, there was an idea that, the bacteria, if you will, sort of, for lack of a better term, knew where to make the right mutations. And so, I mean, that was a fundamental discovery. I mean, they won the Nobel Prize. Uh, I think that's a critical thing. And I often use it in teaching, right? There's a tendency, when you're teaching about it or talking about it, particularly to lay audiences, to talk about, well, the virus mutates to do something, or the virus evolves to do something. And that can convey, I mean, it's shorthand, but it can convey a purpose to the process, that it's, has a direction, and that's not the case, right? I mean, mutations happen wherever, and then selection happens, and most mutations are selected against, and they're dead enders. They never go anywhere, and it's only a precious few that actually confer an advantage and that can get selected for in evolution. And so I found that fascinating, and it shaped a lot of my early work in, when I set up my own research program, because there was this idea, I think, common, that RNA viruses, because they are so successful and they have high mutation rates, and so, boy, it's really a good thing because it allows them to adapt and evolve. But it's a very tenuous lifestyle, if you will, that because, as you say, most mutations are bad. The thing I say is, well, it's easier to screw something up than make something better, and that's what mutations are. And most mutations are very dangerous and are actually lethal to the virus, and very few of them actually make it a better virus or make it better at doing what viruses do. And that was fascinating to me.
Oh, it,
I often analogize. There's this old, I'm old enough that The Simpsons were what I watched growing up, and there's this clip of in one of the shows of Homer Simpson walking around his yard and stepping on rakes. And when he steps on the rake, it pops him in the head. And that's how I think of viruses a lot, is just see, like, Homer Simpson walking around the yard, stepping on rakes and getting hit in the head. Every now and then they're successful, but most of the time it's just that.
I think that when I have discussions with people about
Yeah.
this issue, they can't really when I say comprehend, it sounds like a
Yeah.
value judgment. They
Yeah.
can't really internalize
Right.
the idea of the numbers involved
Yeah.
that
Right.
allow this to happen
Right.
because we're not used to that.
Yeah.
We think, for example, that having 6 or 7 billion people on the planet is a huge number, and it
Right.
is from our perspective.
Yeah.
But I grow greater numbers of that in a culture tube overnight of
Yeah.
bacteria every day.
Right. Right. And that's an excellent point. And I call it it's a numbers game, right?
Mm.
And I think what you point out is both the issues here, and also why bacteria or phage or viruses, so eukaryotic viruses or viruses that infect non-bacteria, um, are such a great system, to do experiments with, because you can generate billions, you know, no sweat, right? It's easy to do. Um, but yeah, at the same time, I mean, I think viruses, it's very much a numbers game. The analogy I use is in a slide often in my talks, is that viruses are like, if you don't like Homer Simpson, they're like people playing a slot machine.
Mhm.
And, most of the time, you're playing the slot, you're pulling the lever, and you come up empty and you've lost your dollar, right? Every now and then you hit the jackpot. Um, that's pretty rare, right? Because the house rules. But when you hit the jackpot, lights go off, the lights go, flashing noises, everyone pays attention, right?
Yes.
And that you pay attention to that. And that's kind of like, influenza or SARS-CoV-2. And, you're like, oh my gosh, the virus did this. That's amazing that the virus did this. Look what it's doing to our society. These viruses are crazy. They're powerful, but that's a rare event. And with SARS-CoV-2, right? It's, hundreds of millions of people got infected, each one of them with, you know, tens of millions of viruses in them. You're going to have a chance for rare events to happen. And yeah, that is a very challenging thing to think about.
I've heard it said, in fact, that when you look just at E. coli in our gut, there are enough of them with
Yeah,
random mutation rates to have
right.
any possible mutation in the genome taking place
Yeah,
many times each day.
right.
And to some degree, those shock and awe statistics are just, I call it stone dorm room thinking, in
Yeah,
that you just think that's really amazing. And it is,
yeah,
but it's also true and it's important to remember. But I'm getting off
yeah.
topic. I have to apologize.
Well,
Um,
if I may, before we go on, I think
sure.
it's an important thing, right? Like, because yeah, the E. coli, every mutation exists in you potentially, right? And people will talk about flu or HIV the same way, right? You have a genome of 10,000 bases. You've got four possible mutations at each base. That's 40,000 possible mutations. If you have more than 40,000 genomes in you replicating, you're going to generate every possible mutation every day, right? So, but I think then that opens the question of, well, why don't we see all of those mutations? Right? Like they're happening all the time, but a lot of them are dead enders. Some of them just never get a chance to make it, to outcompete the other ones, because they're just randomly lost from the population. That's fascinating, right? Like, and I think it begs the question of, if viruses have all this awesome power, how come we're not seeing, we should be dead, right? But they face constraints, right? They face challenges too. And we can learn a lot by understanding that and understanding the constraints. And maybe we can work those constraints to our favor sometimes.
So what I was going to say is, just for listeners and viewers, every virus has some type of genome, and it's
Right,
so interesting to teach this to students because they'll want to know there's one set of rules, and in fact, there aren't.
right,
Constants aren't, variables don't, like my physics
right,
TA told me. But the truth of the matter is, you can have DNA viruses that are double-stranded or single-stranded. And you can have RNA viruses that are double stranded and
right,
single stranded. And there are wonderful examples of each of these.
right.
But I thought it might be really fun to talk a little bit about one that fits the season, influenza.
Sure. Yeah, so as you say, there's all sorts of different kinds of viruses, and they have different lifestyles, right? And we talk about them by the type of their genome and the strandedness. And flu is an interesting one for a number of reasons. It's an RNA virus, so it has RNA as its genome. It's a single-stranded RNA virus, so it's just a single strand of RNA in each of its parts of its genome. It is a negative sense RNA. So there are plus sense and negative sense. Plus sense means that the orientation of the RNA is such that it is 5 prime to 3 prime, and that's how the protein is translated. Poliovirus is a plus stranded RNA virus. Flu is a negative stranded RNA virus, so its genome is opposite to the way RNA works and messenger RNA works. So it's a negative sense single-stranded RNA virus. And then if that weren't enough, it's segmented. So its genome is not just a strand of RNA, it has 8 strands of RNA. And those are like chromosomes. And that endows it with certain biological properties that are really fascinating from an evolution point of view. And we'll probably talk about you know, the importance of reassortment, which is when those get shuffled around. So in addition to mutation, where you make a base change in one of those RNA molecules as it's getting copied, there's a process called reassortment, which is akin to recombination, where you kind of, right, we're used to DNA where you kind of, meiosis, where you have recombination, where you kind of swap parts of a DNA molecule. And that happens, but not in flu. In flu, it swaps information by, swapping those RNAs. So if you have two different viruses infecting the same cell with different ones, sometimes they don't sort properly and they go to the other virus. And so you've shuffled the deck and you have a new combination of RNAs together, which endows the virus with new phenotypes. And that's something that we see with flu. And it's a process called antigenic shift. And I think, I have a slide that, I use in my teaching set that, shows that process of antigenic shift, which is so important for understanding flu and pandemics.
It's interesting you bring this up because these are things that I have taught about and I
Yeah,
cringe as because you know so much more about it than, and I have no false modesty. It is absolutely true that I am not an expert in this field, but when
yeah,
I teach about it, I talk a lot about plus and minus strand.
yeah,
My favorite question that students say to me
yeah,
is they'll say, "Why are there any single-stranded RNA viruses?" And I always respond that that's a theological question more than anything else. And
yeah,
and that's me being flippant, but there are clearly some kinds of advantages to this diversity in form.
right,
But a positive strand virus can act pretty much, as you say, like a messenger RNA and
right,
use the host machinery to make its
right,
proteins and assembly. The
right.
negative strand has a problem because it requires a particular enzyme that
Yep,
our cells do not make,
exactly.
which turns that one strand of RNA into another strand of RNA.
Right.
And
And
so
that's
it,
that's
go
that's
ahead.
a key. I'm glad you're teaching your students because that's fundamental in virology, right? Is like you take poliovirus, which is a plus-stranded RNA virus. I started my career working on poliovirus. That RNA is infectious. If that RNA gets in your cells, you can make a poliovirus because it just gets translated into proteins and then it's off to the races. But as you say, a virus like influenza, it needs that virion, the actual viral particle, needs to have its polymerase, its enzyme that copies the genome. And so it infects the cell and it brings in the tools it needs to make more of itself. And that's a big division in the virus world. Viruses that need to do that and viruses that don't. And then to your other point about, why RNA? Well, right. I mean, you're more expert than me than this, but there's, the idea that life started in an RNA world, and that, this might have been the precursor to DNA,
You,
and that this we're very anthrocentric, right, or DNA centric, right? That we think it's DNA is the fundamental, but it might have been RNA, and so
it's
that's
so
kind of fitting with the viruses.
It's so interesting when
Yeah,
people talk about the origin of viruses, and
yeah,
there are three different models, and I'll
right,
teach those three different
yeah.
models, and students will say, "Well, which one is correct?" And I'll say, "Yes,"
I bet the
because
students love that.
they laugh, but
Yeah,
it's also true. I do think it happened multiple times, and I do
yeah,
think that we don't understand anything about how all those things happened.
right,
And so as a result, especially when you have these viruses that have to, and I use the expression pack their own lunch when they have to take an enzyme
yeah,
with them. HIV does that with reverse transcriptase.
yep,
And it's interesting because then you come into the chicken and the egg argument.
right,
And the answer to that one is yes again, isn't it? But
yeah,
the fact of the matter is this creates a situation where we also need to talk about RNA viruses and I want to say this very carefully because it's
Yeah,
not altogether true. They aren't as careful in their error checking as DNA
right.
polymerases.
Yep. And I think it's an excellent segue. I think so. RNA viruses in general, almost entirely, are sloppy. And this would be RNA viruses like flu and polio, measles virus, mumps virus, hepatitis, A virus. The list goes on and on. Um, Norovirus. They have an RNA polymerase, which copies RNA into RNA as part of as how they replicate. Those viruses do not have, those polymerases don't have a proofreading or repair activities. So they make a mutation that just happens. And so they make, a lot of mutations, one or two, or sometimes three, as we've estimated, every time they copy their genome, which is staggering, right? If most mutations, to your watch analogy, that's a bad way to live. And so, but there are a few RNA viruses that have proofreading. So the nidoviruses, of which coronaviruses are part of that family, um, have a proofreading uh, called a exonuclease, which can actually, excise an error and then restart. And so as a result, coronaviruses like SARS-CoV-2 have approximately a tenfold lower biochemical mutation rate. So you put in a cell, you measure how many mutations, it's about tenfold lower. And so it's closer to it's not quite as low as a DNA, organism like E. coli, but it is lower. And one idea is that this was important in order to have a larger genome. Because if you look across domains of life, the number of mutations per genome is roughly the same, no matter what you're looking at. And so it's called Eigen's paradox for viruses. And the idea is that in order for a virus to have a larger genome, it needed to, quote, figure out how to have a lower mutation rate. And that allowed it to have a larger genome with that, still the same number of mutations per genome, but you could have a bigger template. And so coronaviruses have about a 30 kb genome, which is over twice the size of the flu genome. And if you have a bigger genome, you can code for more proteins. You have, you're more of a Swiss Army knife instead of a single blade. And so that is one of the interesting things. And then you mentioned DNA viruses, right? The important thing to remember is DNA viruses are not a monolith. They don't all have low mutation rates. There are some DNA viruses with higher mutation rates than others, and some of them use different cellular polymerases, right? So there's different error rates of different cellular DNA polymerases, and that can affect the error rate of the virus itself. So tremendous diversity when you start looking closely at mutation rates across viruses.
It's interesting you say this, and it's really
Yeah,
important for listeners and viewers to appreciate this.
yeah,
We tend to put everything into groups. We bin things, for lack
yep,
of a better term.
yeah,
And so we think that things are much more similar than they really are, and we
yep,
don't appreciate the diversity of what's
yep,
taking place. So I'm not trying to say that every organism is completely
yeah,
different, but I do, for example,
yeah,
often warn students that not all bacteria act like E. coli, and
right.
even very smart scientists will suffer from what I call coli centricity. And I'm not making fun of them because it
I
works
got a few of them
really
in my department, some
yeah.
holocentric people.
But the reason is it works so well for E. coli.
Yeah,
And I even had Esther Angard on the show recently, and she made the comment that even E. coli isn't like E. coli because there's
right.
so many different types.
Yeah,
And it's frustrating to students, but it gives me such an appreciation for
yeah,
the beauty of life.
Yeah. We
So,
in medicine we call it the lumpers and the splitters,
yes,
right? There's
lumpers and splitters.
you're faced with this, complexity, and some people make sense of it by, putting things together and into groups, but of course those are very arbitrary and they don't capture everything, but they're useful. And some people are like, no, I want to endlessly divide them to really understand how everything is different, and they both work.
And I get into this quite often,
Yeah,
especially with some of friends we have in common, because
yeah.
people will start changing the names of particular taxons among the bacteria.
I know who
And
you're talking about.
yes, you do. And I realize when I get irritable about this, it's just my impending,
Yeah,
senile nature.
yeah,
I want it the way it used to be. Well, it
yeah.
never is the way it used to be. Is the point?
I mean, you could
So,
even go further, right, into philosophy, right? There's like people who are, deductive and like to kind of break things down to the smallest thing, and then there's people who, more inductive and think about the whole, right, and how, the whole system works. It's just different science.
So let's take a redirect a
Yeah,
little bit
yeah,
here. So when our body encounters the influenza virus,
yeah,
and again, feel free to disagree with me on
yeah,
this. The way that I understand it is the parts of the virion,
yep,
the package containing the genetic information that
yeah,
are exposed to the bloodstream or what your immune system perceives.
yep,
And so that's going to be the fulcrum of natural selection, because if you can avoid being recognized by the immune system,
hmm,
you have something good going on for you from the point of view of a virus. So,
right,
with that in mind, we have something that happens every year,
yeah,
epidemics.
yep,
And people really don't like that term, but epidemic has a relatively innocuous definition.
yeah.
And
It's
the reason that we need to get new influenza vaccines every year is because of, for lack of a better term, this principle of drift.
yeah. Great. I mean, I think it's a great entree into a lot of things to talk about. So, first, I think my evolution colleagues uh, will hate the name antigenic drift because it conjures up a genetic drift, which is a kind of a random evolutionary force, where mutations change in frequency just due to chance, that it's not anything important. The example of genetic drift is, right, you know the native populations, populating the new world, or so-called new world, in the Americas, however many years ago, crossing the Bering Land Bridge, right? And so that meant that, the gene pool derives from what we would consider a relatively small number of individuals, and that's genetic drift. So, it's not because those genes that people brought with them many years ago across the Bering Land Bridge are actually favored or selected. It's just chance. Like they were the ones who made it across, and they're the ones who, um, you know, uh, are the ancestors of these populations. And that happens with viruses a lot, right? But antigenic drift, which we want to talk about, is actually natural selection. And so, as you point out, and I think I have on this slide, influenza viruses have two proteins on their on the viral surface. One's called hemagglutinin, which is what allows the virus to get into our cells. And one is called neuraminidase, which largely functions by helping the virus get out of the cell. And these are on the surface, they're juicy targets on the surface of the virus for our immune system, for our antibodies. And what happens is, and I show this in this other slide, is this antigenic drift. And so that is when, the virus is randomly making mutations. Okay, it's not trying to make a mutation in the hemagglutinin, but it does from time to time. And what'll happen is, we see flu viruses every year, because, you know, they're everywhere and we get infected with them. And every year there's an epidemic, as you say. Where I live, it's between December and February, and you see flu cases go up. And So we build up immunity to those viruses over time. Every now and then the virus makes a mutation that allows it to escape our antibodies. And that is selected. That virus now has an advantage. It can spread better because it's not getting blocked by the antibodies of the herd, of the entire population of humans in the community. And so it starts to spread because it's evading that immunity. And that's called antigenic drift. And often that's due to mutations in the hemagglutinin, sometimes due to mutations in the neuraminidase, which is also targeted by our antibodies, but not as much as hemagglutinin. And so that is why, sometimes we need to update the flu vaccine. And that's why flu vaccination is kind of an annual event. And SARS-CoV-2 or COVID vaccines might, become an annual event as well, because, that's a process that the virus undergoes. But I'll just say one more thing and then I'll shut up on this is it's important to recognize, right? It's not just about making the mutations, right? It's about how those mutations are able to spread. Uh, for instance, you look at measles virus. Uh, we've had the same measles vaccine for what, you know, 4 or 5 decades. It hasn't changed. The virus has a high mutation rate. It's an RNA virus. It makes mistakes, but for whatever reasons, we're still figuring out antigenic drift has not been a problem with measles virus, and that's fascinating.
And
Um,
thank
and
goodness.
and RSV, respiratory syncytial virus, also makes a lot of mutations. We have a new vaccine. Um, I think we're optimistic that it's not going to have antigenic drift. We don't know. And so there's a lot to learn about why some viruses do this and some viruses don't, because it's not just about their ability to make mutations.
No, and I think
Yeah,
something that we've talked about in the podcast many times
yeah,
is the interaction of different things that
yeah,
we tend to be reductionists in our
yeah,
view.
right.
So this now, by the way, for listeners and viewers, if you've ever wondered where the nomenclature for a lot of influenza viruses comes from,
Yeah,
like H1N1,
yeah,
now you know.
yeah. I think I have a slide, influenza virus ecology, right? And there's gosh, I'm going to say the wrong thing. I think there's 18 or maybe 19 HA types. My flu people will get very upset at me for not knowing this, and something like 10 or 12 NA types. And so there's a lot of potential combinations there. And yes, so the seasonal ones that we deal with are H3N2, which has been around in us since 1968, and H1N1, the current version, has been around in us since 2009. And why those dates? Well, those were influenza pandemics, and that kind of gets to your prior discussion about antigenic shift and reassortment. And what happens is there's a huge reservoir of flu viruses that are in largely, bird populations. Some are in bats, but almost entirely influenza A viruses are in birds. And what happens is there's this mixing and matching, and then sometimes they get into mammals, and maybe there's other mixing and matching. So pigs are a big one, but there's other mammals that will get infected from birds, and then sometimes it'll spread to us. And so when that happens, you have this completely new looking virus. Its H and N's are completely different, and we don't have immunity. And that's when you get a pandemic. And so the 1968 is when H3N2 crossed over. Um, 2009 is H1N1. And notably, there was a H1N1 before, and this one pushed that H1N1 out because it was different enough. And so a lot of the flu world is trying to figure out, is there going to be another one of these events? It's not if, it's kind of when, and being prepared. And so right now, your listeners have probably heard about H5N1, and that is, another kind of combination. And of course, in the U.S., there's been this issue with H5N1 has apparently gotten from birds into dairy herds of cows, and so there's cows infected with H5N1. And there's been, sea mammals infected by H5N1, and so there's this spillover that's happening increasingly into mammals, and we're mammals, so we got to pay attention to it.
I'm really glad that you brought up what my students, and they actually say it sounds like a dance, the shift and drift. But
Yeah,
in fact, both of these things are important.
yeah,
Epidemic, pandemic. My late parents had been through several of these
yeah,
pandemics, and my great grandparents through, of course, the big one in 1918.
yeah,
But these things are, as you say, always going to happen.
right.
And so it's important to be able to find ways to fight them in some
Yep.
ways. Some folks are working on antivirals, improving the way that we make vaccines. But the part that I think I want everyone to appreciate, especially in the current climate, is that because the viruses have the ability to change their I'm inferring intentionality. Because the viruses change their surfaces,
Yeah,
they those particular variants are able to avoid the immune surveillance that a vaccine can give us, and it
right.
makes it very, very tough.
Yeah, it does. And I often think when I teach this, I try to get the point across that antigenic drift and antigenic shift don't happen in isolation, right? I mean, they're all happening all the time. And so you take something like H5N1, so it has gone under the shift, but it's still evolving through drift. And maybe not to evade our immune systems. Maybe it's to, it's affecting its ability to, infect a cow, uh, you know, uh, and get into milk, and or it's being selected for that. And then there's also mutations in the virus that make it better able to replicate. Maybe virus mutations in its polymerase that help it work better in a mammalian cell than in a bird cell. And there's all sorts of differences. And the virus is always adapting. Or maybe it's you throw these H5 and N1 together and they don't work well together. They're a new combination, and so it maybe undergoes additional changes that make those two proteins work well together. So there's just a lot going on, and like you said, it's always going on.
I think it's important to
Yeah,
remember. And the other part, and one of the things that I worry about is the negative imagery we have about this. The truth of the matter is we're constantly swimming through a sea of microbes, including viruses, all
yeah,
the time.
yeah.
And we have this idea that we're coated in Purell,
Right,
and we're not. And
right.
it's we co-evolved with so much of
Yeah,
this.
yeah,
So I think it's an important paradigm that we need to keep in mind, because you can't get away from this is my point.
right,
It's
right.
going to happen regardless.
And then I think kind of to build on your point a little bit, I mean, of course, there's, the I guess concept or theme of One Health, right?
Yes.
That you have, these viruses that are in animals, and we need to be good stewards of nature and domestic and wild animals, and kind of understand this in order to protect ourselves, protect the animals. I mean, and it's not just for the good of principles, right? It's agriculture. The chicken economy is huge. As we've seen, the dairy economy is huge, and we need to understand how our practices and how our interactions with animals make this more or less likely, and what we can do about it for the good of animals, the good of ourselves as well.
I mean, if you think about it, we can, in the laboratory, evolve microbes,
Yeah,
specific microbes, to live better under particular conditions than
right,
normal.
right.
I am quite sure that the majority of our lab rat microbes well, I know they've diverged quite a bit from their more wild type relatives.
Yeah,
So this idea that we live in a vacuum is ridiculous, and it's
yeah,
inaccurate. I hate
right.
to be so dogmatic, but it's
Yeah,
also true.
Right.
And at the same time, look at the problems that there are always going to be problems in mass
Right.
production of anything. And that's what
Right.
we're dealing with here.
Right. Yeah.
So the people that come up with the flu vaccines have to make guesses
Right.
about
Yeah. I
possible
don't think
changes.
anyone would like calling it a guess. uh, educated guess. I think, right? We always like to think that evolution is a predictive science, right? It's not. I mean, we're working toward that goal, but it's
Right,
not. And so I think the where things are now is a more of a kind of surveillance regime with evolution layered on top. And so what I
right,
mean by that is in flu for a number of years, um, and now with SARS-CoV-2, and now with RSV and other viruses, what happens is there's an enormous effort put into sampling and sequencing viruses from all over the world. We participate in that in my lab for all those agents, many labs around the country and around the world. And the idea is, then you have a real-time picture of what the virus is doing. And then you take the epidemiologic and evolutionary modelers who look at that data, and then they could say, well, this one is seems to be growing faster and spreading faster. And we look at it, and then we add these other data, and it's got mutations here. We know these mutations will do this. Let's test it. How it responds to antibodies, and all that's done year round, all the time. And then everyone gets together, the experts, and they look at the data and they're like, well, we're a little concerned about this, and this looks like it's going to be a problem. And so we think when we make the next formulation of the vaccine, it should have that a representation of that virus in the vaccine. And that's what they do. And this afternoon there's a meeting, for RSV vaccine where we're looking at some of our data on RSV and saying, has the virus changed in any way such that the vaccine needs to be reformulated. And so that work goes on, and so it's data driven, and it's based on a collaborative investigation that's global, because it's so important to global health.
two things really quickly
Yeah,
is the first is when I took epidemiology
yeah,
about a gazillion years ago,
yeah.
uh, my professor told me that the job of an epidemiologist is supposed to be wrong and people are happy about it. And that's true
Yeah,
because, and then I see this a lot in our modern, round, quick, kind of news economy that we've
yeah,
got, or news ecology that we're
yeah,
creating.
yeah.
And you have to make your best, I guess is not the right word, your best
Prediction,
prediction. Yeah, prediction that is going to happen.
hypothesis.
And I have seen situations, and I don't have them
Yeah,
off the top of my head, where the particular influenza vaccine isn't a great fit for the
right.
circulating virus.
Yeah,
And then people lose their you know what over this. And
right.
I'm thinking, listen, you have to make
Yeah,
predictions about what's going to happen and get ahead of the game. And it's not going to be
yeah.
100%.
I'll add something else which might be of interest to your listeners is because it's evolution. Um, so as folks may know, the vast majority of flu vaccines that are made are grown in eggs. Okay, and so you come up, flu virus grows very well in eggs, and so it's been a time-honored method for making large amounts of flu vaccine every year. Now, it's great. Um, but there's other ways of making flu vaccines, and some are made in mammalian cells, some are recombinant, et cetera. And so, but what can happen is you pick the virus for the vaccine, you put it in the eggs, you grow it up in mass quantities in production, and guess what? Sometimes the virus will evolve. And so it will have, quote, egg adaptations. And so it's sort of like antigenic drift. And because sometimes the virus in that process will make a change in its hemagglutinin that changes the way it looks to our immune system. And so you could pick the right strain, but sometimes when it grows up in eggs, it undergoes a change. And so the antibodies that you make might not protect you against what's actually being spread in people. And so it's another layer of challenge here. And a lot of the quote vaccine, uh, mistakes in choosing the strain can come from these changes in eggs. And so a big area of research is looking for alternative platforms, cell-grown vaccines, mRNA vaccines, things like that that can, uh, you know, circumvent this problem.
I'm not surprised at
Yeah,
all. I mean, the hand
yeah,
of Darwin is on everything, and there's
yeah,
a really good example of it.
yeah, for
I
sure.
had friends a long time ago, Adam, who were doing bacterial transformations sequentially, and they would pick the very first colonies that came up because they were in a hurry. And invariably, they were super mucoid because you saw them first
Yeah, yeah,
and they weren't the best choice. So they
yeah.
were a selective agent.
Yeah, or in my case, it's because the bacteria has kicked out the plasmid that you're trying to grow up, and that's why
Yeah.
it grows faster because it doesn't have the plasmid. And so, yeah, always pick a variety of sizes. That's the lesson to teach any molecular biologist.
Parenthetically, you had an amusing image that you saw from down the street. Would you care to share that with the
Oh
listeners
yeah,
and viewers?
so You know, I'm blessed. I get to walk to work most days or ride my bike, even year round here in Michigan. And there's this place near my house that's on my way in that used to be a coffee shop, many failed businesses, but now it's a new business of cannabis, which is now legal in Michigan. And in a bizarre way, all these places have unusual names, but this one's called House of Evolution. So I really like that, and I liked making that. I thought that would be an appropriate theme for my lab as the House of Evolution, and so I took a picture of it, and I now use it on my slides. And so I think we have a slide of it here. And so I also consider it free advertising for them now. That
Oh my.
I don't know if there's an overlap in my
Yeah.
audiences and their customer base.
Past the Oreos. So what we've done today is we've talked about a
Yeah,
lot of stuff.
yeah,
And I appreciate how we talked about how mutation works a little bit, that it's
yep,
random. We talked about how some different viruses handle different aspects of that. We talked about RNA viruses and spent some time with influenza. And we even talked about the way that vaccines might work. There's just one thing more, because time is growing
yeah.
short, that I'm curious about. Do you have any thought about antivirals for influenza?
I do. Um, I think they're good. No, I mean, I think antivirals have had it's tricky, and they're not miracle drugs, but it's an important part of our armamentarium. And the challenge is, right? Look, the flu only lasts a few days, and so a clinician will look at it as like, well, what benefit is the patient going to have from this? And to really get the maximum benefit, you have to treat someone early. And you know, even then, some people might quibble about the benefit. Um, and but I think they're good. I think they help people. And it's between you and your doctor to interpret the data and discuss it. I think they're good, and I think there's benefits to the patient and to the community with antivirals. But yeah, I put my money on vaccines in terms of kind of reducing the burden of disease for a lot of these viruses.
I know many people have talked about antivirals, and
Yeah.
clearly you have to look for a particular target.
Yep.
And for RNA viruses in general, that RNA directed RNA polymerase
Yeah.
would seem like a decent target. I know
Right.
many people are working on that.
Yep. For sure. I mean, it's a great target. It's very distinct from our proteins. It's, as they say, more easily druggable. You're able to develop a compound that can target it. without as many side effects. So yeah,
Adam, with quote optimal matching
yeah,
between a particular strain of influenza going by and a vaccine,
yeah,
when you actually look at the numbers, what I have read is that it's not quite as effective as some of the effects of, for example,
right,
the messenger RNA vaccines against SARS-CoV-2.
right.
Is there a reason for that, and what can we do to fix it?
Yeah, I think this is going to sound like a dodge, but it's not. Uh, I'm not an epidemiologist, but I hang out with a lot of them and participate in a lot of these studies of vaccines. And it's interesting. Uh, it's all about how you measure. Right? Okay, when you say it's effective, what's it effective against? So in clinical world, it's what's your endpoint? Are you measuring how well the vaccine protects you from infection? How well
Mhm.
it protects you from getting a symptomatic infection? How well it's protecting you from going to the doctor? Or how well it's protecting you from landing in the hospital or dying, right? Those are very different benchmarks, right? So that's one issue, and comparing the numbers, it's challenging. Then the other challenge is, right? You think about it, it's like, okay, well, I'm going to compare the vaccine against so people who got the vaccine and people who didn't, right? And who does better, and how well does it prevent people from getting, whatever outcome. That the COVID vaccines look great early on, right? Just in that study, right? But what happened is a lot of people have gotten infected. Almost everyone has had COVID by now. And so then when you do the study again, so they have some immunity, right? And so when you do the study again, you compare the COVID vaccine against people who didn't get the COVID vaccine. All of a sudden, the difference is going to be less because the people who had COVID before still have some immunity. And so you're comparing two things that are closer together in terms of that person's protection. And so the vaccine doesn't look as good. You take something like flu, where we've all been assaulted with flu viruses our entire lives, either by getting infected or by getting an annual flu vaccine. And there's actually, you know, complicated immunity and complicated epidemiology that affects how susceptible we are in any given season. And so that can make it look worse in terms of how much protection you're getting the vaccine if you just look at that top line number. But it really has to do with a lot with how the studies are being done. And I wouldn't be surprised if you just looked at the top line numbers with SARS-CoV-2 or COVID vaccines over time, those are going to drop, but it's not because the vaccines aren't working. It's because, our comparator group isn't that different in terms of the background immunity. And so the importance becomes just maintaining that immunity. And as we've seen with SARS-CoV-2, immunity goes down over time. Your body forgets that it saw something, and so we need to boost it. And that's kind of the logic here. And so, entire careers are spent looking at these subtle differences in measuring how well vaccines work.
I don't think it was a
Yeah,
dodge at all. I think that how you define the
yeah,
terms is very
yeah,
important. I'm really glad that you
yeah,
you said that. I think it's
yeah,
helpful to a lot of people. Thank you.
sure.
Adam, I've also heard from time to time people, and I'm going to use the term bandy about the idea of a universal influenza
Yeah.
vaccine. Now, of course, the minute I say universal and any living thing, I get really worried. What are your thoughts on this particular subject?
Yeah, I think it's a very important area of research, and there's a lot of good ideas out there. And so the challenge is sometimes people are talking about different things when they're talking about a universal flu vaccine. One would be, let's say you had a flu vaccine that worked against all H3N2s. Okay, we talked about how there's different H and N types of flu. The so you like, okay, that way maybe you don't need an annual vaccine against H3N2 or H1N1 because the immune response triggered by that vaccine is against all of those and all of the possibilities. So you are done with H3N2 or H1N1. Um, so that's one type of universal vaccine. Another type is to say we're going to protect you from all flus, right? So you design a vaccine that is going to provide protection against not just H3N2, H1N1, but will provide protection against H5N1 if that ever becomes an issue, H7N9, and target parts of the virus that really will confer broad protection. And that's a big challenge. But yeah, I think we got to go for it. I mean, I think it's important, and there's a lot of scientific hurdles or interesting scientific problems to solve along the way, which will help us in general. And so, I'm all in favor of us working toward that goal.
I often say that you need to find
Yeah,
the Achilles heel
yeah,
immunologically. And
yeah,
and if you have some kind of target,
yeah,
antigenic target, that if it evades that kind of fit
yeah,
to an immunoglobulin, to an antibody, that
yeah.
maybe it no longer works the way it should, then that's
Right.
something we're thinking about. But evolution is sneaky.
Yep. Yep. Right. It's sneaky, and it's like, well, right. We'll never be sure we're done. Right. You can come
Right.
up with a universal vaccine, but you got to keep looking and making sure that it's still working, because there's always the chance that things will. Who's the guy in the Jurassic Park? Nature will find the way. Right.
I actually, that's true. And I actually have one that it was from Francis Crick quoted it. And I think it was Leslie Orgel who said that evolution is smarter than you are.
Right.
And
Yep.
it's really true.
Yep.
So I want to be clear that especially when you talk about influenza, there's
Yeah.
so many directions that we go. I'm going to
Yeah.
put some links into the show notes,
Yeah.
and perhaps in a future episode of this podcast, we'll get a historian on to talk about the
Yeah.
1918 flu. That would be
Oh yeah.
a
Yeah. This
wonderful
great,
topic.
great books written on that. It's a
Uh,
fascinating.
And you know what? Many over the years, it's so
Yeah.
interesting to me.
Yeah.
But what I was going to say is I really appreciate your time.
Yeah.
I just had one more question
Yeah.
for you. Was there any one incident that convinced you had to be a microbial scientist? It's okay if you don't have one.
Yeah. I mean, I'll just tell you how I got into it. I think it's so I was in my second year of or my first year of that MD PhD program at the University of Washington in Seattle, and up until that point I was very interested in molecular and cell biology. I had worked in labs studying yeast and signaling in yeast, and I was very interested in cancer
Mhm.
and cell cycle, and I just thought that was the best thing in the world to study. Um, and then I went to a lecture, and it was Julie Overbaugh, was at the University of Washington and now is at the Fred Hutch Cancer Center in Seattle. And she gave a talk about retroviruses and feline leukemia virus. And she studied this, mostly it as a model system for HIV. It was an early model for HIV. But she gave a talk and talked about how this virus would cause tumors in cats. And it did this by altering signaling. And I thought that was just fascinating. And so I was not interested in viruses at all, but that started me down the road of understanding retroviruses, how they evolve, how that's linked to how they cause disease. That was my PhD work on feline leukemia virus, and that just kind of got me hooked. And I came out of that lab card carrying, microbiologist, virologist, and I never looked back. And so I I was fortunate to go to that lecture that day. And Julia has been a fantastic mentor to me my entire career.
We need to get cards that say that, don't you
Yeah,
think? So we can be card carrying microbiologists.
yeah, for sure.
Adam, it's been a pleasure chatting
Yeah,
with you today. I had a wonderful time chatting with you before the podcast started. I hope we can talk again in the
yeah,
future. And
yeah.
I want to wish you and your family and your colleagues the very best.
Well, thank you. This has been a tremendous amount of fun, and thank you for all you do in terms of education and outreach about, microbes. It's very important.
You're very kind. Thanks again.
Take care.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with a lot of links of interest, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Adam Lauring is at the University of Michigan with appointments in the medical school and microbiology and immunology. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.